also recording this. Feel free to ask us questions anytime. I mean, ask John questions anytime. And we take questions on Facebook as well. So go ahead, John. Thank you.
Okay. Well, welcome, everybody. I can't see you, so I don't know how many of you are out there or what you're doing right now, but welcome to the Time Machine, Museum Time Machine. Yeah, I have been at Idaho State University for 11 years. And I had come from another job in California to come out here. And I've had the extreme luck and privilege since I've been here at Idaho State to be able to put together some laboratory infrastructure and equipment for use by everybody, but mostly we cater to archaeologists and people who do paleo science, paleo environmental kind of things. And so I'll be talking about some of that as I talk about the things that we do with teeth today. And so feel free to ask any questions. And I've asked Amber to just go ahead and interrupt when a question comes in, so we can talk about those things. So yeah, so I would consider myself a let me just share my PowerPoint. I would consider myself a bioarchaeologist, strictly speaking. And for those of you who don't know what that means, it's somebody who really looks at the physical, organic, and inorganic characteristics of the human skeleton and oftentimes animals that accompany people in the archaeological record. And so that's kind of what I do. And we use a lot of technology within our attempts to do bioarchaeology. There's bioarchaeology that can be done at a very kind of a macro scale, where you look at the skeleton and you look at the way that bodies are interred, and some of this kind of goes over into mortuary archaeology as well. But the kind of the bioarchaeology that we do is, I usually call it microbioarchaeology or molecular bioarchaeology, because we like to look at the very small pieces of things that attend the human skeleton and kind of stick to us, stick on us, stick in us, either as a feature of life or something that happens after we die and we're buried. And so these things can be really illustrative of particular processes that have happened both in the life of the individual and the kind of things that happen post-depositionally. This is a field of study called taphonomy, what happens to the skeleton after it's buried. And so this is where most of the applications that myself and my colleagues and my students use to understand the archaeological record in this way. So today I'm going to be talking about kind of some case studies of things that we do in the lab and why I think they're important and maybe telling for the purpose of why we would do this kind of bioarchaeology and what it can inform us about. So again, the topic that I'm talking about today is teeth, and teeth as time capsules. In the upper left here, you can see there's a human skull, and the inorganic remains of the human body, once it decomposes, is usually comprised of the skeleton. Now, the skeleton's not entirely inorganic. There's a lot of organic materials that are stored within the skeleton, but we generally conceive of the biominerals, the actual bone material, as a kind of an inorganic material. And when you look at it in electron microscopy, you can see that there are kind of orientations of things. These are both teeth in the upper right and the lower left. These are electron micrographs of teeth showing you the kinds of structures that occur in teeth. On the upper right is the prismatic elements of the enamel, and on the lower left is showing you the kind of the hollowed out structure of the dentin, which is like the body or the root of the tooth, and these little tubules that go through it that show you that communicate material in and out of the tooth as the tooth is in a living condition. On the lower right, there's a model of a tooth here, and this is just to give you a brief orientation of what we're talking about here. Now, you can see that the top of the tooth is covered by enamel. Underneath that, you have a material called dentin, which is a bony material. It's very much like bone in any other part of your body. The enamel is quite different. The enamel has very much less organic material in it, and it's a hard protective coating on the surface of the tooth. Its primary function is to let you chew things, right? And then there are some organic, very organic features of the tooth, and that includes the pulp chamber of the tooth, which is the kind of the inner portion of the tooth, which has a blood supply and nerves. On the edges of the margin of the root of the tooth, you have ligament attachments that attach the tooth to the bony socket that it's in your mouth. And there's a material that kind of lines the body of the root on the outside of the root called cementum, which is also probably another one of these measures to hold the tooth securely in place within the jaw when it's in either within the mandible or the maxilla, the upper jaw or the lower jaw of the mouth. So this gives you a little bit of an orientation. Teeth are interesting because they're very hard, and teeth tend to be some of the hardest bones in the body. The dentin portion of the tooth is some of the hardest bones in the body, and it's also interesting in that most of the skeleton of an organism is in a constant process of erosion and reformation. And so any of you who have ever broken a bone know that the bone knits back together, and if you wait long enough and you get an x-ray of it, the doctor would look at it and said, I can't tell that you had a broken bone there. And that's because the bone remodels through time. What's cool about the teeth is that they don't remodel through time. They form one time. Well, they form twice, actually, because you have a set of baby teeth, your deciduous dentition, and then you get a set of permanent teeth, so you get two separate tooth formation events. But the tooth itself only forms one time. And so this is interesting and good for archaeology because, one, it can kind of lock a record in of what was happening during that time of formation. The other feature of it is because it's hard material, the dentin is a hard material, it can preserve as good or better than most other components of the skeleton once the individual is interred, put in the ground. And so this gives us a really good ability to find these materials. And sometimes in archaeological sites, you'll find loose teeth, but you won't find very much of anything else. And that's a testament to, one, the kind of the smallness, discreteness of the tooth, and two, the fact that it is a little bit harder and a little bit more protected than some of the other skeleton, either in the skull or the postcranial skeleton. So this is why we like to look at teeth. Teeth are good records for a lot of interesting things in archaeology. Now, teeth are, again, your primary means of interfacing with the world of the things that you eat. Your teeth are, these very hard, very conserved elements that are designed to allow you to chew, to bite, to strip, to do things with, to get food into your mouth. And some people use their teeth as tools as well. But we know that when we look at teeth, and when a dentist looks at a tooth, he sees something maybe a little bit different than what us as archaeologists sees. He sees pathologies, things that need to be corrected. He sees tartar or dental calculus that needs to be scraped off. He sees, periodontal disease happening with the ligaments of the teeth as it attaches to the gums. Archaeologists don't think about it in that way. We see all of these things as a record of events that happened in an individual's life that can tell us things or help us understand things about the life of that individual, or alternatively about what happens to an individual after it's buried. So, a dentist looks at your mouth and says, I see a lot of problems here. And an archaeologist, a bioarchaeologist, will look at your mouth and say, hey, I see a lot of interesting information here that we can glean from these materials. So just briefly, and I'm not going to go into this in any great detail, biomineralized tissues, such as bone and tooth dentin and enamel, are all based upon an inorganic mineralized structure. It is modally what we call calcium hydroxyapatite. And the formula for it is there if you want to know what that is. It also includes the residues of the formation process, the amyloblasts and osteoblasts, which are these kind of bone and enamel forming cells. And we also have in teeth the remnant of the first formation of the tooth, which is this kind of collagen scaffold, or this kind of framework with which the tooth mineralizes around. And collagen is a kind of a rubbery material that subsequently becomes locked in by mineralization. The non-mineral portion of biological appetite, non-mineralized organic portion, varies from around 2 to 5% in dental enamel and about 20 to 25% in dentin. Now, in cortical bone and like all the other bones of your body, it's 30% plus of this organic material. And so it gives you another idea about why maybe in some cases the bones don't preserve maybe as well as these more highly mineralized portions of the skeleton, the teeth, and the dentin of the tooth and the enamel of the tooth can. So that's one of the reasons also why teeth tend to have maybe some higher survivorship.
John,
in our lab, go ahead.
Before you move on, we have a question from Xavier. He would like to know, how long do teeth stay in the mouth of an animal? So I think, when do animals maybe lose their baby teeth?
You know, I'm not super familiar with that, and I don't actually, in fact, know if all animals have a deciduous dentition. I would imagine they do. These baby teeth. Dogs, for instance, which is what I know, will grow their first set of teeth and then basically chew them out over the course of a few months, probably beginning when they're maybe like 4 to 6 to 7 months old. I had a dog when I was a kid that had its adult teeth come in, but it didn't lose any of its baby teeth. And so it had double the number of teeth that a mouse should have. And it looked particularly ghastly. This dog did not look very healthy at all until we took him to the vet and had these supernumerary teeth removed, the deciduous teeth taken out. So, yeah, I don't really know very much about when most animals would tend to lose their teeth, but I think that's something we could probably look up and probably get a little bit of understanding of, maybe in Wikipedia or something like that. So our lab's primary areas of research include looking at elements of diet, looking at maybe the isotopes of stable isotopes of plant materials and animal materials that an individual would eat. And these can get stored into the dentition. And as I said before, because the dentin of the tooth only forms one time, the bone of the tooth only forms one time, it can lock in that information of what kinds of things you are eating during the time of the development of those teeth. And as a matter of fact, there are some people, and we've had a couple people here in the lab who've tried this, who look at a tooth, and you can see in the upper right here, there's an electron microscope image of a tooth. The gray area here is dentin, the more wider area is the enamel on the top of the tooth. What these researchers did is they sectioned the tooth in increments all the way down. We cut through the tooth, cut right through the middle of the body of the tooth, all the way down from the cusp down to the root tip, and looked at the isotopes of the different kind of plant materials plant and animal materials that they were eating, so we could actually get kind of a timing of life of how the diet would change as the tooth was forming. As your permanent dentition is forming, when you're just a couple of years old, your permanent dentition is already racing along in your body, getting ready to pop out when you are between 5, 6, 7 years old is when your permanent teeth start to come out. Those teeth are already forming, and so that can lock in a signal of the kinds of things that you're eating when you're young. And then if you think about the very tip of the root, which is the last part that forms, that may be anywhere up to your early 20s if you have your wisdom teeth that are forming during that time period. So you can get a nice long look at the diet of an individual. The other thing we look at is these micro residues that get trapped in the dental tartar, or again, what we call dental calculus, that can give us information about the kind of specific things that people put in their mouths. We also look at things like life history exposure. And this is something I kind of coined, this idea that if during the time of your life you are exposed to a lot of things, some of the things are very good, nutritious foods, high mineral diets, things like this, and some of the things are particularly bad, exposure to toxic metals or other kinds of intoxicants. And these things can become lodged into the tooth and can give us an estimation of the kinds of exposure events that have happened, especially for people in the past. And that'll be one of my case studies. Also, we like to look at taphonomy, the process of how a tooth changes after it's been buried. Post-depositional alteration is an insult that happens to bones. It can change them dramatically. In acidic environments, it can actually completely remove the bone material. So all you're left with is kind of a gooey former presence of an individual. In very alkaline environments, very base-like environments, you can have a perfectly preserved skeleton that has absolutely no organic material left in it. These are kind of things that we're interested in because we want to know what are the limits of what kind of preservation can happen. And then something I'm not really going to talk about today, but something we've been doing a lot more of, is skeletal forensic analysis, especially with teeth. And we've been working with some local law enforcement agencies on some cold case ventures to try to understand or identify potential individuals from cold cases and some of the kind of things that evidences that would lead us to potentially solving that case. And I can't really go into it much more than that, but these are some of the things we look at. So the first case study I wanted to offer up to you is one that I worked on several years ago, and it's becoming more and more interesting all the time. It's called Metal Production in Early Bronze Age Ural Mountains. And so the upper left image here is a aerial photograph of a village. It's called Bar 5 with an associated cemetery with it. And this is a Bronze Age settlement. Meaning that it's in Europe that occurs anywhere from about 4,000 to about maybe 2,500 years ago throughout Europe. And Bar 5 is really interesting because it's a fairly well preserved village remain, but it's also a really well preserved cemetery as well. And some of the interesting things about it is this is an example of this, a culture called the Sintashta culture, which was prominent in this area around 4,600 years ago. They made chariots. They had there's probably some of the people who domesticated the horse. They pulled chariots. They had very complicated burials, very kind of very ritualistic burials. And in the upper left, you see an image here. This is a burial that has a chariot on the bottom that's been buried, and then above that it's been laid over with a material, and then above that the individual's been buried, been laid over with a material, and then above that there's animals, horses that have been put in there, and then oftentimes there's feasting remains that are in these cemetery vaults as well. Lots of really great artifacts that come from these things, pottery, things like pipes, spear points, early metal spear points, lots of grave goods, really interesting stuff. One of the things that you do find in these particular burials is there is the remnants of the bronze culture. And these are some of the very first bronze makers in the world. And what's interesting about that is that they were using bronze that was a kind of a self-made bronze. Today we think of bronze as a mixture of copper and tin. But there are bronzes that exist that in these early metal making cultures that were made out of copper that had native amounts of arsenic in it, anywhere from I think it's about 1 to about 5% arsenic in it. And when you melt that down and you melt the ore down and you make a metal artifact from it, it makes this very pretty kind of almost a silvery looking bronze. And it has a great feature to it that it almost never corrodes. So even bronze artifacts over time will get that kind of green patina on them and they'll start to kind of rot away. Arsenical bronze artifacts persist a lot longer. But for those of you who know anything about metals and you know what arsenic is a potentially very poisonous metal. And so the thing that really came out from this is that when my colleague was excavating this cemetery, he found out there was a bunch of kids in the cemetery. And really more kids than you would expect to see in a cemetery if it was a kind of a normally operating population. It just looked like there was a lot of kids there. And so the speculation was, are these people committing infanticide or killing off the youth? Was there some kind of a disease that went through? And so he approached me about looking at some of these materials and seeing if we could identify any evidence of potentially even metal intoxication, arsenic intoxication. And the reason that they thought this was because when you make this kind of arsenical bronze, You have to heat up the ore in a very hot fire. It releases tremendous amount of a gas called arsenious trioxide, which just floats out of the atmosphere. It's available to be breathed in, and it can build up in the body. And so the way that they had identified these kind of not the burial, but the village sites, it looked like the houses would have been these kind of low earthen kind of affairs where they would have a relatively small area, perhaps maybe 15 to 18 feet long, maybe about 10 feet wide, with a low roof on it, and with a fire pit in one corner where they were making their bronze. And they even find molds of metal artifacts in some of these house excavations. And so the idea is that if you're making bronze in a low house like this, and you're living in the Ural Mountains where it's cold and windy, and you're staying inside to keep out of the weather, you're ingesting tremendous amounts of arsenic gas. And so this was what they thought maybe one of the things that was yielding this higher rate of mortality in the child population. So they gave us a whole bunch of bone materials, and we went through and we sampled them, and we have a laser ablation system, and that's what this upper right here. It's basically a high-powered microscope with a laser that shoots out of it that can drill into materials and then take the material that kind of is whisked off of it as it is being augured out, and take it and analyze it with a mass spectrometer. And so this could get us all the metals or all the inorganic elements within a matrix, and these are these bone matrices that you're seeing here, and these are the sampling sites that we sampled along them, and we saw a tremendous amount of arsenic. But the problem was we didn't know if the arsenic was coming from the soil, because these people are buried with these metal artifacts, lots of metal artifacts, or if it was something that was actually built up in the body during the life of the individual. So this is, again, this idea of what happens during life or what happens after death. These are the kind of questions. And so when we look at this isn't even actually that important, but it's showing you that some of the distribution of the metals that we saw in the bones themselves. And what we see here is this is on the top is iron, below that is manganese, and then below that is arsenic. And we can see that on the surface of the bone, there's this kind of patina that's developed here. And that is the bone that's exposed to the dirt. As the body decays, the dirt exposes and comes right up against the bone. Certain of these mobile elements in the soil, things like iron and manganese, transfer very easily. They kind of stick on the surface of the bone. It gives the bones this kind of a brownish black appearance. But the arsenic looked like it was going all the way through. And so the question was, is the arsenic just traversing right through the bone, or is it something that is being was lodged in the bone during the time of life, something that we can take a look at using this method? And so we decided what we had to really do was look at teeth, because again, as I said before, teeth are harder, they're more impervious, they tend to preserve better, and they preserve the signal better of what happens to them. And so we did some analyses on some teeth, and this is an example here of a tooth here that has been lasered into over 900 times on a very small scale here. And what we did is we plotted the elements that were inside of the enamel and in the dentin of the tooth. And below here is what you see. This is manganese on the left, arsenic, barium, and uranium. And the reason we picked these elements is because these elements do different things. They move differently when somebody's buried. As you can see here for both barium and uranium, both of these look like they're coming in through the pulp chamber, through the open space in the center of the tooth here, and they're kind of migrating from that area out towards the top of the tooth and the enamel. Barium does it quickly. Strontium, the metal strontium, does it even more quickly. Uranium, which is like barium and strontium, does it a little bit more slowly, but you can see that it's moving into the pulp chamber, the roof of the pulp chamber of the tooth. Now, we figured that arsenic might behave somewhat differently than the barium and the uranium would, and might behave a little bit more like one of these metals like manganese or iron that kind of plates the exterior surface of a tooth and starts building up and moving in, but at a much slower rate than barium or uranium. And so as you can see on the left-hand picture here, the manganese, you can see this very dark distribution of manganese. This means that the manganese is completely kind of plated the exterior of the tooth and is starting to move into the enamel just a little bit. But if we look at arsenic, we can see that there is a little bit of that happening on the surface of the tooth, but really not very much. And there is no presence of arsenic that is, kind of like a wave moving into it, like you see here with the barium and the uranium in the middle of the tooth. And what this meant to us is that we're probably seeing arsenic building up in the teeth that's actually in proportion to the arsenic that was taken up during life. And that in fact, what was happening is that in this village, they were making arsenical bronze from arsenic-rich copper ore, and they were making these beautiful tools. But at the same time, they were probably really poisoning their family. And the kind of justification we came up with this is that when you are young, there's a period of time where you're kind of at risk. Your liver is not fully functioning to remove toxic metals or other substances as you get into your body. And so these things can become lodged in the body. Whereas when you become an adult, you have a fully functioning liver, you have a bigger body mass, you can spread out that metal, that poisonous metal a little bit more evenly in your body, so that when we see the evidence of these children that are at very high proportion in the cemetery, we think that maybe what was happening is that by the process of making these metals, they were probably poisoning everybody, but the children are most susceptible to it, and that's why they show up at more frequently in the cemetery than you would expect from a normal population. So it's kind of a cool, not really a detective story, but a story about how you could look at the occupation of somebody. This person is making metal, they're living on the steppes of Russian steppes, and they are doing the best that they can. 4,600 years ago, they're making beautiful artifacts, making beautiful weapons and other kinds of metal objects. But at the same time, they're causing a real disastrous outcome potentially to their family members. This last part here is just, we looked at the arsenic concentrations in people who work in smelting operations today and compared it to the amount that we found within our sample. And the arsenic that we find is fully consistent with some of the higher exposures that modern people can get the exposures that people were getting archaeologically about the same time. Second case study is organic biomolecule survival in the Western Pacific. And so this is one of the places that I work more than just about any place else. And when we look at the Pacific, we realize that the Pacific is marked by being mostly in tropics and subtropics. It's in the ocean, so it's wetter, it's warmer, and we expect the survivorship of all kinds of things not to be as good as they might be in a more northern area. For those of you who know about this, you can still go to Siberia today and they're unearthing mammoths in the permafrost that look like they're just a dried out, fully perfect preserved mammoth. And that's because the cold and the dryness of those areas tends to create a lot of preservation. The Pacific is just the opposite. Warm and wet with lots of sun exposure is really hard on these kinds of materials. And so oftentimes we don't see very good preservation of skeletal materials in the Pacific. And so part of our analysis is to study at what level these things become disturbed or altered to the point where they're not really very usable anymore for doing these kinds of biomolecular studies. And the three places we look at, and I'm going to go over this fairly quickly, is in the upper left here is a place called Saipan, which is in the Northern Mariana Islands of Micronesia. We also have the site of Taumako, which is in the Duff Islands, which is a Polynesian outlier group north of Samoa. And then we have, not north of Samoa, it's well further, it's north of Melanesia. And then we have Fiji, which is the island of Fiji, which is down here in the lower right. And these are all from different time periods. Taumako is relatively recent, 700 to 300 years ago. Saipan, probably in the neighborhood of about 900 years ago. And then Fiji is significantly older, up to almost 2,000 years ago. The environs of these were in the site of Saipan, was a beach dune burial. with a bunch of skeletons that were interred. The preservation of the skeletons outwardly looks very good, but they're buried in this very kind of sandy beach area. And so the preservation of the biomolecules is not great. Again, as I mentioned before, you can get really good preservation of the bone, but maybe not preservation of the organic materials inside of them. Fiji, this is a place called the Kotoka Dunes in Fiji, and these are burials within a sand dune. Again, a sand burial, very different kind of sand, a geological sand versus a sand derived from coral and shells, but also a very kind of a harsh environment for skeletons, and really resulted in generally speaking, very poor preservation of organic materials. And these are some historical photographs of unearthing these skeletons from these beach dunes. And so we use this tool called an FTIR tool that allows us to look at the molecular components that are remnant in bone and tooth. And in this case, we just used purely dentin from teeth. And that's because we could control for that across all the different materials, all the different sites we were looking at, and really compare survivorship of dentin, the organic materials in dentin. And this graph below here is just a graph showing you what we look for, the kind of peaks that we look for. And it may be a little bit hard to see here, but there's a red line here that shows you what a modern tooth would look like. And then there's a blue line which shows you what an archaeological tooth would look like. And so the major differences we see is that this peak here, which is at 1640, you don't need to know that, is called the amide peak, or what collagen is made out of. And collagen, again, is the primary organic material that's resident in a tooth, in the dentin of a tooth. And what we see here is that as things are buried and this process of taphonomic alteration takes place, these organic molecules get taken away, and there's some replacement that happens in some of the other molecules. So these are crystalline molecules of the hydroxyapatite here. But what the takeaway picture of this is that this gives us a really good look at the amounts or the levels of preservation we're seeing in the organic molecules in these materials, and we can compare them through time. I really don't need to talk about this very much. This just talks a little bit about what we see in terms of the phosphate. Calcium hydroxyapatite is a calcium phosphate, and there's also a component of that that's made out of this thing called bone carbonate. And then one of the measurements we take is something called the splitting factor. You don't really need to know. I'm not going to go into that in any great detail. You can look that up or review that. So what we see in our experimental data is that as these materials become more altered, we see a reduction in the amount of this so-called amide to phosphate ratio. So this is really the collagen to the most resistant part of the bone of the tooth, which is the phosphate molecule. And we can compare that to this thing called the splitting factor. And we can see that through time, you'll see a distribution of effects that take place on a tooth. And so this is all the data put together here, all the samples put together here. Now, this is the interesting graph here, because this shows you what the preservation looks like in these materials by different kinds of class of materials for what they are. And so this box here is modern samples. These are teeth that the dentist extracted from somebody's mouth, either because it was diseased or broken or something like that, and then they were given to us as experimental samples. And so this shows you the amount of collagen that's in them, and then this very low splitting factor, which is really a kind of a reorganization of the crystalline matrix of the tooth. Both of these things can tell us something about the preservation and maybe time as well. And so if we look at the samples here and the samples that are potentially the best preserved here, these Taumako samples, which are potentially only about 350 to about 700 years old, tend to have a lot more of the collagen that's remnant or that's left in them. The Saipan samples buried in the kind of the calcareous dune area of the Mariana Islands have a lot less of the collagen in them, and they have this higher splitting factor, this greater reorganization of the crystals. When we look at the Kotoka dune samples, the mass of them occurs up here. So almost no collagen left in those samples, and also the highest amount of crystalline transformation of those the dentinal crystals within it, the hydroxyapatite crystals. So this gives us a picture of what kinds of preservation we might expect to see in any sample from the Pacific, based upon knowing that, you know, the kind of the temperature regimes, the amount of rainfall, the water regimes, the conditions of burial, and the age of the samples really can all come together, interplay together in a significant way to help us understand why certain things can be preserved or not preserved. And again, collagen is one of the main materials that is recovered if you want to understand the diet of an individual, looking at stable isotopes of carbon and nitrogen and sulfur. You can look at the collagen that you can recover from it and get a probabilistic idea of the diet of an individual, the kinds of classes of things that they're eating. Are they eating a lot of plants, a lot of plants that have kind of a grass signal in them, like corn? Are they eating a lot of fish versus a lot of terrestrial animals? These are the kind of things that the stable isotope profile can help you understand. The big issue is, can you get enough collagen, and is the collagen degraded to the point where you might not get a very representative look at the diet of an individual? So that's why we conduct studies like this. We also look at the presence of collagen for looking at the presence of DNA that can be recovered from a sample. And so these are samples that we expect to have. This is just another view of this data here, but showing you where we're trying to recover ancient DNA from these samples, because we believe that if there's presence of collagen in the sample, collagen occurs at high frequency, there may be the presence of ancient DNA left in the sample. And so this is the kind of the way we're looking at this. And so we looked at across. What you see on the right here is this is a gel image, and so this is showing you the amount of DNA that is recovered and can be amplified in a PCR reaction from some very degraded samples. And you can just barely see the very faint bands here that show us that we're able to recover DNA signal out of samples that are varying levels of of alteration. The positive control here is a modern DNA sample, and so this shows you that the reaction worked. The extraction blank, which is a negative sample, shows you that it isn't in that sample, but the other four here, 1, 2, 3, 4, show you that there is DNA that was recovered from these. A very little bit, but there was recovered from some of these, and some of them didn't recover any DNA at all. And if you look at the tooth samples here on the left-hand side, these are samples from Saipan. They're in horrific kind of preservation condition. They're very crumbly, dry. We really didn't expect to recover anything from these samples, but yet we were able to get material out of them. The Fiji samples, being significantly older, actually were in even worse condition, and very little DNA was recovered from these samples when we did this analysis. And so this was predicted by the collagen that was remnant in samples, this amide to phosphate ratio, which can give us some clarity going forward if we're going to look at a sample and try to extract material from it. Which one would we pick? And this can help us screen these samples very quickly to identify potentially samples that we would prefer over other samples when we're looking at human teeth. This also shows you the collagen recovery here, percentage of collagen recovery, and whether DNA was recovered or not from samples. And so you can see that there's not a perfect one-to-one comparison here. In this one here, we have 9% collagen recovered from the total weight of the material that we analyzed. And in both of those cases, the 9%ers had DNA in them. But as you can see here, there was one here from Saipan that looked like the visual inspection and preservation was poor. We got a lot of collagen out of it, but we didn't get any DNA. Here's an example from Fiji where the condition we visually observed was fair, the collagen was very low, and we didn't get any DNA out of it. So it's not exactly one-to-one, but it is the kind of thing that these studies can help us build up kind of an observational and an empirical assessment of what we expect to find in the samples going forward. The last case study I talk about is the study of diet from Easter Islands from the Easter Islanders, the Rapa Nui people of Easter Island, that we were able to look at using dental calculus. This is a picture of some of the samples that we recovered here, and you see these bands here. This is right where the gum comes to meet the tooth, and normally say, well, you shouldn't be able to see any bone. You can see this is the line of the enamel here, and then this is the dentin of the tooth, the actual body and root of the tooth here. Normally, the gum comes up right to the edge of the enamel, or very close to it. But in certain cases where the oral health is not particularly great, you can get the gum resorbing a little bit and showing a little bit more of this dentin here. And this area right along here is where the dental calculus tends to build up. So a dentist might look at this and say, I could see there's some pretty evident pathologies going on in the mouth of the individual who had these teeth, based upon the kind of the gum resorption, or the way that the gum is receding away from the tooth, and then the presence of this dental calculus, which most dentists think plays a part in oral disease and early tooth loss. But this is a gold mine for archaeology, because these bands of material here, this hardened kind of biofilm material that forms on these teeth at this margin here, can hold on to a lot of interesting things that go through the mouth of the individual. So in Easter Island, the kind of plants that people eat, or the plants that we think that people ate in the past, there's some modern kind of remnants of them today. And they built these subterranean gardens. These are called Monavai gardens in Easter Island. And this is because the island is whipped by pretty ferocious winds during parts of the year. It's definitely subtropical there, and you get a lot of salt spray that comes off the ocean. And unless you can protect your plants, the plants generally get whipped by the wind, and then they also get salted by the wind, which causes the plants to be stunted or die. And so what we see is a lot of these subterranean gardens with plots with things like taro and sweet potato, root crops that are growing, and probably also gourds would be growing in these things as well. Actually, that's tobacco right there. But in these gardens, we see the remnants of what we think were the dietary staples of the people of Easter Island. This on the upper left here is taro. This is taro that's just been pulled out of the ground before being cleaned up. This on the lower right here is sugarcane. This here in the upper middle is the sweet potato. And then there's some evidence of banana in Easter Island, although banana is tough to grow there because it is subtropical. But we do see remnant evidence of all of these materials on the island. When we look at them microscopically, we can see that there are pieces of these materials that get preserved. And these are 4 cases here that you can see. On the upper left here, these are pollens from a palm tree. The right here, you can see there's a bunch of things in this image here, this upper right image. But really what I'm focusing on is this kind of spiky ball here. This is what's called a phytolith, or a plant skeleton. And this plant skeleton comes from a palm tree. It's called globular echinate. It's very commonly identified to palm. The lower left, you could see these kind of crosses that you can see in the image here. And I'll show you a little bit better images here in a minute. These are the polarization product of looking at starches. Starches are a polymer, and when you polarize the light on them, they'll show this cross. It's called a Maltese Cross. It's usually very indicative of starches. And then on the lower right here, these are these little spines that show up in taro. It's also spines you see in lots of other plants. Rhubarb has it in it as well. These are called calcium oxalates, or rhaphides. They're these little spiky things that grow in the plant. They're probably a defense mechanism for the plant. If you eat them without processing them, you can get hurt. And that's why people say don't eat rhubarb unless you cook it down. Same thing for taro. If you eat taro raw, you'll blister your throat. You'll probably end up going to the emergency room. And so these are the kind of things that we might see. These are both inorganic materials here. The pollen and the starches would be the organic component. These are all potential targets to see in dental calculus. And here, these are just some of the examples what we see. This is banana phytoliths from dental calculus. These little dog bones here are sugarcane phytolith. And then this is a phytolith that comes from a gourd. plant. This isn't our work, this is from another author. But these are the kind of things that we might expect to see in the calculus. Other things we see in the calculus, which are often odd, are things like cysts that are kind of waterborne, kind of plants that grow. Diatoms, any fresh water that pools up on a surface and exposed to sunlight will grow these things in them. And then if you drink that water, you can subsequently incorporate them into the tartar in your teeth. Starch grains, here's a good example of this Maltese Cross here. This is the light microscope image of it. And so here's the Maltese Cross here. Here's the yam, starch without the polarized light on it. This is a banana, and so forth. So we go looking for these things in the calculus. And as you dissolve the calculus, you can recover these materials. So these hardened bands of tartar on the teeth, we usually dissolve them. We initially were dissolving them with acid, and that proves not to be the best for the survivorship of other things. And so now we do it with a kind of a decalcifying solution. But we would record the calculus on the teeth. We would do the extractions in a clean room. And this is super important because these phytoliths and starches and pollens blow through the air. They're so small, they can contaminate everything. And so we have to do it in a clean room environment. We also did modern exemplars as well to show that we could recover these things in modern materials and that we could have a key, a visual identification key to be able to observe them in the archaeological materials. So this is a sweet potatoes skin being boiled off in nitric acid in order to recover the materials within them. We would visualize them in a scanning electron microscope. This is our microscope in the lab. And these are the kind of things that we see here. I'm going to go through these pictures fairly quickly, but you can get just a sense of the kind of the beauty of these very small things. These are down in the neighborhood of 20 microns, 5 to 20 microns in size. So it's 5 to 20 millionths of a meter in size. They can be recovered at high frequency. Again, these are the bilobates. So these are the grasses, probably from sugar canes that show up in dental calculus. Conical phytoliths, these are probably, I don't know exactly what these are, but I won't speculate, but these can be attributed to specific genuses of plants in order for us to identify what moved through the mouth. The diatoms were very interesting to us in this study because we showed that there was so many diatoms that were packed into the dental calculus that it really gave us a perspective that probably most of the water that was being consumed by people on the island was water that was sitting on the surface. So not like a spring flowing from underground, but probably rainwater caught in catchments or in shallow lakes, brackish water supplies. These are some of the clusters of diatoms, truly amazing masses of these things all stuck together. Cysts as well, which shows that there's other organisms in these waters. And then things like little sponges and other kinds of organisms. Starch grains, we identified some starch grains. The interesting thing is that the predominant thing that we found other than diatoms was these globular echinate palm phytoliths. And that was very odd to us because it really made us think that maybe they were eating the trees. And it was so kind of alarming from that standpoint. And of course, the popular speculation about Easter Island is that people went down and they cut down all the trees and they drove their society into chaos and cannibalism. But what we really see is that the sediment, the actual dirt of the island, is so loaded with these things, these plant skeletons from palm trees, that the island must have had an enormous history of being covered by palm trees prior to the arrival of the Easter Islanders getting to the island. And so in a bit of a detective story, my former student and I went ahead and took sweet potato samples from various locations and actually boiled them down and found loads of these palm phytoliths in the sweet potato skin. And then we actually were able to actually, this is sweet potato skin here in this image that's just been broken. It's been frozen and broken. And as you look at it, you could see these little globular echinate phytoliths, these little spikes are stuck into the skin of the sweet potato. And so it really solved this is really a detective story. It solved the problem where we initially thought, well, people are eating the palm trees on Easter Island and incorporating it into their mouths. we think, well, no, actually what's happening is that they're eating a lot of tubers, sweet potatoes, and probably taro, and they're incorporating it into the calculus by chewing on it. You cook a sweet potato in a fire, and it still has the skin on it. You eat it. The sweet potato doesn't give you any phytoliths, any plant skeletons. It gives you starches, but no plant skeletons. But yet, because it grew through soil that was really rich in these plant skeletons from palm trees, this was a secondary means of incorporation. So it was a really kind of a clever study that my student did to be able to prove this, and that was a lot of fun and a good study that came out of it. The last thing I'll talk about very briefly, just frontiers-wise, is last year we published an article looking at the occurrence of this blue pigmenting agent, which turned out to be lapis lazuli, which is a semi-precious gemstone that showed up in the calculus of nuns working in a monastery in Germany about 1100 years ago. And this very brilliant blue pigment showed up. And so we did a bunch of analysis of it in order to prove what it was, that it was actually lapis, and probably lapis coming from Afghanistan. Some of the earliest occurrence of the trade of these Singh semi-precious stones from the East into Central Europe and Germany. And the story that's told from this, and it's not exactly clear what the actual final story is, but it's either one, these are nuns that are illuminating manuscripts. They're actually going in and painting in the manuscripts with different pigments to make them more beautiful, so that they're brought to your attention, and that potentially that just licking the brush as you're painting it with these materials incorporated into your teeth. There's other potential explanations for this as well, but this is what we think is the most probable. So it's actually another good example of the kind of things that you can learn even in maybe a slightly more modern context from the context that I normally work in, that can help you solve these kinds of problems about occupation, diet, what people are doing that lead these things to be preserved in the body, and especially in the mouth. And as we like to say, the mouth is the orifice where everything comes in. It's where your water, your food comes into your body. It's also definitely could be, your teeth can be used as tools. Lots of people use their teeth as tools. So it's really a primary way to interface with the world around you. And so that's why we study teeth, and that's why we study the things that both are in teeth and attach themselves onto teeth, because it can really give us an insight into the daily activities in a way that just looking at the skeleton or looking at the artifacts strewn around an archaeological site can't. And so that's why we find it really interesting. So that's really all I had. If there's any questions, I'd be happy to entertain them.
Yes, you actually have a couple. Xavier would also like to know, do the teeth you have that you find, do they have different hardnesses?
That's a good question. It's not really a hardness thing. It's more of a resiliency thing. And so it would be the difference between like a piece of marble, the stone marble, and a piece of chalk and a sugar cube. Now, if you hit all three of those with a hammer, each one of those is going to do something different, right? The sugar cube is probably going to completely disintegrate into individual granules of sugar. The chalk might break up into bigger chunks. The marble, the hammer might just bounce off of it. Those are the kind of hardnesses that we talk about. And it's really not so much as hardness as is how coherent is the material? Does it stay together? So that's a good question. Yes, we find lots of differences. Sometimes you'll pick up a piece of tooth and you'll say, this might be a good candidate. And as you're holding it in your fingers, it literally, you crush it and it turns into powder. And then you realize, well, that probably is not a very good place for us to start with. Maybe we should look for a better one.
And then Xavier's mom would like to know, does a single tooth give enough information to be useful, or are numerous teeth necessary?
Single teeth are really ideal. Sometimes it's good to have multiple samples of teeth, but really what we are looking for is sometimes in an archaeological excavation, you'll just find a tooth. And sometimes the tooth is such good shape, it's like, you wonder, did somebody just pry that out of their mouth and throw it over their shoulder next to their camp? Or is this something from a skeleton that got dislodged and then something took the skeleton away afterwards? So sometimes it's just these individual finds, and those can be very useful to us. And mostly when we work with people, we ask them, could you send us a couple of tooth samples so that if one of them doesn't pan out, we always have another one to go back to. But we've done a tremendous amount of work on individual teeth, and especially individual teeth that if they have dental tartar, which is calculus on them, if they have enough material for us to take a sample of the root for a stable isotope analysis to look at the diet of an individual, and if there is enough material that's left over for us to do a DNA analysis. One tooth can lead us to at least 3 different pieces of data, and potentially more than that. There's a lot of frontiers out there of what people are doing with dental calculus and different ways that people are looking at the organic materials in the dentin of the tooth that are really kind of fascinating and unique in a lot of ways. It really gives you an idea of the kind of the ingenuity of archaeologists to study these things at ever smaller scales.
Well, if anyone else has any questions, now's the time to type them. That was really fascinating, John.
Thank you.
It was a fun look around the world. And solving the palm issue in Easter Island, that's a great detective story.
I could talk about that for hours. It was just so interesting. We spent so much time on it, and every time we got more data back on it, it kept getting weirder and weirder. And we actually looked at nuts, the actual, because this tree makes a coconut, really small one, and we looked at the nut itself to say, is there any of these plant skeletons in there? Because we were thinking, would they have, cut down the tree and used the pulp of the tree itself? Because I don't know if people know, you can make palm flour. So palm tree trunk, you can actually grind it up and make a flour out of it. That's nutritious, and we actually make pancakes with palm flour. And we're thinking, were they eating the flower of the tree? And then we looked at the pulp, and it didn't have those plant skeletons in it. It's really the leaves that had the plant skeletons in it. We're like, well, okay, how could we think of what they were using leaves? Were they stripping the leaves and making baskets out of them? Were they actually eating the leaves, which is impossible because you couldn't get any nutrition from that? How was this happening? And it wasn't until we actually really started looking at the sweet potato that we figured out, yeah, it's growing in the soil. And then we had some dirt samples from Easter Island, and we looked at those in the microscope, and there was more plant skeletons than there was dirt. Literally, there was 75% of the mass of the dirt. You get a handful of dirt in your hand and you say, this is just dirt. 75% of it was plant skeletons. The rest of it was what we would normally consider dirt. That's how many are on the island. And so that sealed the deal for us and really enabled us to talk about the story in a more complicated way.
Xavier has another question. What is the most complex tooth you have ever found?
I would say it's the teeth from Russia, the teeth from the Kamenye Ambar 5 cemetery, because those had a lot of calculus on them. We didn't actually look at the calculus. We sent that back to the investigator to have him look at it, and then he wanted somebody else to look at the DNA in it. But really understanding the way that these metals were moving around inside the tooth, and being able to pick apart the signal of metal that came into a tooth during the time that the bone was forming, that the tooth was forming, versus what happened after the individual died and was in the ground, was really fascinating. And it was a ton of math, really some complicated models to understand how certain elements would move in and out of the tooth over time, and then the ones that would stick in there. And that was a very complicated kind of story that we were able to tell about that. But I think that was, to my mind, one of the most interesting. Definitely I spent the longest time trying to figure out what was going on with it. So.
We also have a question from Facebook. Pamela asks, can you pinpoint different times where things are taken into the teeth in reference to how old the individual was when they came into contact with those minerals, like reading rings on a tree?
I don't know if we're there yet. I don't know if we have the technology to be able to do that. We have tried to parcel out. I mean, I did a study several years ago, another one of these studies that we did, looking at children's teeth, at deciduous teeth or baby teeth, and it's called the Tooth Fairy Project. and we were looking at metal incorporation, specifically the metal manganese. We were looking at mercury too, but manganese ended up being the one that was the kind of the culprit in children that were diagnosed with ADHD or autism spectrum disorders, and comparing them to a control group of children. And these are all from Southern California, and really well kind of validated and cross-referenced study, and looking at see if we could find out the timing of maybe an exposure to a certain metal. Manganese is really interesting because it's necessary for the body. You can't have none of it in your body because it's very bad, but you can't have too much of it in your body, and too much of it is at a very low level. And I can't say specifically, several parts per million is too much. And if your body can't filter that out, it can get stored in the teeth, in the deciduous teeth. And so we were actually trying to see if we could see the kind of the daily load that would have occurred as the tooth was forming. We weren't able to get it to that level of specificity. But as I mentioned at the outset, the stable isotope project that happened in the Duff Islands, where researchers sectioned the teeth from the root all the way up to the crown and looked at those individual segments to try to see differences in diet as that tooth was forming. There is ways to do that. There are ways to try to do that. I think that maybe the ways we do it now are a little bit crude, and I think there's some development of method that needs to take place. But I certainly think it's possible.
All right, well, we are at our hour. That was really fun. Thank you for joining us, John. You have a great afternoon, and thank you to everyone on Facebook and on the Zoom webinar that joined us today. Thanks.
Thanks, everybody. Appreciate it.